Assalamu alaikum. Welcome to Wetlag. Today the topic of my discussion is gastrointestinal motility. So before starting our topic, we should know that what are the layers of gastrointestinal tract. If we cut transverse section of gastrointestinal tract, then we see that innermost layer is mucosa. After mucosa, there comes submucosa. After submucosa is muscularis externa, and after muscularis externa, the outermost layer is serosa. The innermost layer, which is mucosa and which also faces the lumen of the gastrointestinal tract, contains epithelial cells and endocrine cells. The next to mucosa is submucosa, and in submucosa there is nerve supply and blood supply. Then comes the muscularis externa. Muscularis externa has outer longitudinal muscles and inner circular muscles. Means it has two parts. Further, muscularis externa is divided into two parts. Its outer part is composed of longitudinal smooth muscles, while its inner part is made up of circular smooth muscles. And at the last, the outermost layer of gastrointestinal tract is serosa, and this functions in lubricating and protection of the gastrointestinal tract. Now let's start our topic, which is gastrointestinal motility. The walls of gastrointestinal tract are composed of smooth muscles, and these walls are capable of movement, and these movements of the gastrointestinal tract act on the ingesta in the lumen of gastrointestinal tract and cause peristalsis and movement of ingesta throughout the gastrointestinal tract. As gastrointestinal wall is composed of smooth muscles, and all smooth muscles within a muscle bundle are connected with each other through gap junctions. And similarly, the smooth muscles present in one muscle bundle are also connected to the smooth muscles of other muscle bundle. Uh, so we can say that smooth muscles which are present in the gastrointestinal wall are interconnected with each other. So any activity in one muscle is passed to the other muscles also. In this way, they act as syncytium. They act as syncytium and are known as unitary smooth muscles. A syncytium means a large structure which is formed by joining two or more cells. So the muscles, smooth muscles which are present in the gastrointestinal wall act as syncytium and form a unit, and therefore they are known as unitary smooth muscles. And because of the gap junctions which are present between the smooth muscles, uh, we can say that all muscles are electrically connected, forming a network. As we know that muscularis externa of the gastrointestinal wall is composed of outer longitudinal and inner circular smooth muscles. There are only few gap junctions between the longitudinal smooth muscles and inner circular smooth muscles. On the other hand, there are more junctions between the longitudinal smooth muscles. So longitudinal smooth muscles are well interconnected between themselves rather than that of circular smooth muscles. So there are many gap junctions between the longitudinal smooth muscles, while between the longitudinal smooth muscles and circular smooth muscles only few gap junctions are present. The gastrointestinal motility is very important because of the several functions that it performs. Some of its functions are that gastrointestinal motility propels the ingesta from one location to the next. So it helps in peristalsis of food. It also causes to retain ingesta at a given site so that it can be digested, absorbed and stored at that particular site. Then movements of gastrointestinal tract also help to break up food material and to mix it with the digestive secretions that are secreted by submucosa. Then the other function of gastrointestinal tract or gastrointestinal motility is to circulate ingesta so that all portions come into contact with absorptive surfaces. So it propels the ingesta throughout the gastrointestinal tract and retains ingesta at those points where the ingesta needs digestion, absorption, or storage. Then it also breaks the ingesta and mixes it with the digestive secretions which are secreted by submucosa and continues to circulate the ingesta so that all the portions of ingesta come in contact with the absorptive surfaces of the gastrointestinal tract. Gastrointestinal motility means the movement of the gut, and this movement or gastrointestinal motility may be of three types. It can be propulsive movement, it can be retentive movement, or it can be mixing movement. In propulsive movement, it propels the ingesta throughout the gastrointestinal tract so that it can be transported to all parts of the gastrointestinal tract, and in this way it helps in peristalsis. Second type of motility is its retentive motility. So as it propels the ingesta, it also causes the retention of ingesta in those parts of gastrointestinal tract where ingesta needs further digestion or absorption or storage for some time. During the digestion of ingesta, it also causes its mixing with the secretions of gastrointestinal tract. So the time that is taken by the gastrointestinal tract to move the material from one portion of the gut to another portion is referred as transit time. So when ingesta passes through the gut instantly, it means that the propulsive motility is increased, because as the propulsion of ingesta is increased, it will take small time to pass through all the portions of gut. On the other hand, when propulsion of the ingesta is decreased, then it increases the transit time. When propulsion of material will be slow, it will automatically take more time to pass through all portions of the gut. So in this way, transit time will increase. So up till now, we have discussed that the gastrointestinal motility is the movement of gut wall, and there are three types of movements of gut walls, that are propulsive movement, retentive movement, and mixing movement. And these three are also the functions of this gastrointestinal motility. That is, it causes the propulsion of food, retention of ingesta, and mixes it with secretions of the submucosal secretions of the gastrointestinal tract. Then we have discussed the composition of gastrointestinal wall, that it is made up of smooth muscles, and there are gap junctions between the smooth muscles of gastrointestinal wall, and it forms unitary smooth muscles and acts as syncytium. Then we have discussed the transit time, and we have discussed that increased propulsion decreases transit time, while decreased propulsion increases the transit time. So now come to the control of gastrointestinal motility. Smooth muscles that compose the walls of gastrointestinal tract have an intrinsic electrical property. And that intrinsic electrical property is that they spontaneously produce waves of partial depolarization. And these waves of partial depolarization sweep over the entire length of gut smooth muscles. So, smooth muscles spontaneously produce waves of partial depolarization. Now, the question is, what is the origin of this electrical activity of smooth muscle cells? There are specialized smooth muscle cells known as interstitial cells of cajal, and they form an interconnecting lattice of the cells, and this lattice surrounds the circular and longitudinal layers of muscles over the entire length of gut. And these interstitial cells of cajal originate the waves of partial depolarization and cause rhythmical and spontaneous oscillations in the smooth muscles. These interstitial cells of cajal are interconnected with one another and also to the smooth muscle layers by tight junctions, and these junctions are plexuses. These plexuses or tight junctions act as connections between the interstitial cells of cajal and the smooth muscles and cause the flow of ions from cell to cell. So this movement of ion from cell to cell through tight junctions or plexuses causes the propagation of waves, and these waves are of partial depolarization and they travel across large number of cells present in the gastrointestinal tract. In the gastrointestinal smooth muscle cells, the baseline membrane potential is usually 70 to 60 millivolts. And under the influence of interstitial cells of cajal, the membrane potential fluctuates from this baseline level by as much as 20 to 30 millivolts. Thus, under certain resting conditions, the depolarization is only partial and membrane potential never reaches 0 millivolts. So, interstitial cells of cajal are responsible for the production of slow waves, and under resting conditions the resting membrane potential of these waves is 60 millivolts to 70 millivolts. And one important thing to notice is that this resting membrane potential is in has negative values, and these are minus 60 millivolts to minus 70 millivolts. So these waves are called partially depolarized waves until their membrane potential reaches 0 millivolts. When their membrane potential will reach 0 millivolt, these waves will become fully depolarized and contraction will occur. These slow waves constantly pass over the gastrointestinal smooth muscle, whether it is actively contracting or not. And one important point to note here is that the slow waves are very important for muscle contraction, but they are not the direct stimuli for contraction. Although these slow waves are constantly passing over the gastrointestinal smooth muscle, but they are not direct stimulus for contraction. They cause contraction to some extent, means that they are important for inducing contraction in the muscle. But these slow waves are not the direct stimulus for contraction. The actual stimulus for contraction is the action or spike potential, and these spike potentials are produced only when there are slow waves. As these slow waves are characterized by incomplete depolarization, the spike potential or action potential, the waves that are produced by this spike potential are characterized by complete depolarization of membrane, and because of this complete depolarization, contraction occurs. So the modulation of these slow waves in order to produce action potential or spike potential is under the control of nervous system, that is enteric nervous system, and endocrine paracrine factors. So this action potential is produced by the modulation of slow waves, and that modulation of slow waves is caused by the enteric nervous system, endocrine and paracrine factors. So, in order to cause the muscle contraction or to produce action potential or spike potential, these neurohumoral regulatory molecules from enteric nervous system's neurons and endocrine paracrine factors are released in the vicinity of the smooth muscle cell, and these neurohumoral factors affect the ion channels that are present in the smooth muscles membrane, and these movements of ions through these channels influences the baseline membrane potential, and this resting potential changes to action potential or spike potential. Now let's repeat it. Neurohumoral regulatory molecules from enteric nervous system and endocrine paracrine cells are responsible for the for influencing the baseline membrane potential, that is resting membrane potential, and to produce action potential or spike potential. So these neurohumoral factors are released in the surroundings of smooth muscle cells, and these neurohumoral factors affect the ion channels that are present in the membranes of smooth muscles, and these ion channels cause the movement of certain ions through them, and then this movement of ions influences highly on the baseline membrane potential or resting membrane potential and changes into action potential or spike potential accordingly. So these neurohumoral factors are composed of excitatory molecules and inhibitory molecules. The excitatory molecules elevate the baseline membrane potential. It means that it brings the resting membrane potential closer to zero and cause contraction. While on the other hand, inhibitory molecules lower the baseline membrane potential and makes it more negative and, and causes inhibition of contraction. So, when the membrane potential of a smooth muscle becomes close to zero, action potential occurs and muscle contracts. Neurohumoral substances that are excitatory cause smooth muscle contraction by elevating the baseline membrane potential or resting membrane potential. On the other hand, inhibitory substances inhibit muscle contraction by lowering the resting membrane potential. And in this way, the contractions of gastrointestinal smooth muscle mass is synchronized by the action of slow waves, enteric nervous system and endocrine paracrine system. The frequency of muscle contraction is synchronized by the frequency of the slow wave. So muscle contraction occurs at a frequency that is exactly similar to the frequency of slow wave. For example, slow waves in the canine's stomach occur about 5 times per minute. The crest of each slow wave in this 5 minutes in 1 minute may or may not produce the action potential. So during a given minute, the muscle in the localized area may not contract at all, or it may contract up to 5 times per minute. So if these passing slow waves generate no action potential, the muscle will not contract at all. And in a given minute, if action potentials are associated with only one slow wave, the muscle contract only once. And if action potentials on two slow waves result in two contractions, and if action potentials are produced on the crest of three slow waves, then three contractions will occur per minute. And if action potentials are produced on four slow waves, then four contractions will occur per minute. And if action potentials are produced on five slow waves, then five contractions will occur per minute, and it is the maximum limit of the contractions per minute in case of canine's stomach. So in short, when slow waves pass over an area of smooth muscle without causing action potential, no contraction will occur. And when slow waves pass over an area of smooth muscle and action potentials are supremosed on slow waves, means that the resting membrane potential changes to 0 millivolts, then gut muscle will contract. Under normal conditions, in the small intestine, changes in membrane potential begin high in the duodenum and are propagated away from the mouth along the length of the small intestine. These aborally moving waves of partial depolarization are called slow waves or basic electrical rhythm of the gut. It means that under normal conditions, in the small intestine aborally, if we move aborally from the small intestine, the changes in the membrane potential begin high in the duodenum and are propagated aborally. And when the changes in the resting membrane potential will be high, there will be the production of more slow waves. So in case of dogs, slow waves occur about 20 times per minute in the small intestine, while in the stomach and colon, slow waves occur less frequently and occur about 5 times per minute. So, up till now, we have discussed that smooth muscle contraction is first stimulated by interstitial cells of cajal. And we have also discussed that slow waves have an important relationship with muscle contraction, but they are not the direct stimulus for the contraction. And we have also discussed that slow waves are constantly passing over gastrointestinal smooth muscle, whether this muscle is actively contracting or not. But we have not discussed the production of resting membrane potential and the spike potential yet. So as we have discussed previously that there are certain channels in the membrane of smooth muscles, and these channels allow the transport of sodium, calcium and potassium in and out of the cell. So when membrane potential is decreased, these sodium and calcium channels will cause their release in the cell, and it will cause an increase in the electrical potential of membrane. And when this electrical potential will reach to its peak, the membrane will be slightly depolarized, not completely depolarized, but it will be slightly depolarized. So when this depolarization will occur, then potassium channels will open and these potassium channels will cause the efflux of potassium, and potassium will move out of the cell, and the electrical potential of the cell will become low due to the loss of positive charge. So due to this loss of positive charge, the membrane will again be repolarized. Then due to this decreased electrical potential, again sodium calcium channels will open and cause the release of sodium and calcium in the cell, and this will again increase the electrical potential of the cell. And when the electrical potential will reach at its peak, the membrane will be slightly depolarized. And when the electrical potential will reach at its peak and membrane will be slightly depolarized, then again potassium channels will open and will call repolarization. So in this way, membrane will be slightly depolarized, then repolarized, then slightly depolarized, then repolarized, and so on. So these fluctuating potentials that are produced in the smooth muscles are known as slow waves, and they are not the action potential. Slow waves are not the action potential, and they don't produce contraction in the smooth muscle. Now I am going to discuss the production of action potential. So before discussing it, you should know that what is threshold potential. Threshold potential is the membrane potential at the point where sodium permeability caused by depolarization becomes just equal to the potassium outward flow volume. It means that threshold potential is a membrane potential of the membrane at that point where the influx of sodium volume becomes just equal to the efflux of potassium volume. As we know that resting membrane potential is minus 60 to minus 70 millivolts, and this resting membrane potential is only changed into action potential when its electrical potential becomes 0 millivolts. During depolarization, the electrical potential of the membrane decreases and it tries to approach the value of 0 millivolts, which is necessary to produce action potential. But as the membrane is only slightly depolarized, so under resting conditions, this slow wave cannot produce the action potential. At resting conditions, the depolarization value of the slow wave is only minus 60 to minus 70 millivolts. But when the depolarization reaches above minus 55 millivolts, then neuron will fire an action potential. So this value of minus 55 millivolts is the threshold potential. And if neuron does not reach this critical threshold level, then no action potential will fire. So when the peak of slow wave touches the threshold potential, then voltage sensitive sodium channels will open and will cause a lot of calcium to trickle in the membrane, and membrane is depolarized. So potassium will move out due to this activity, and it again becomes repolarized and comes back to its original condition. And if slow wave is still at threshold, then it again depolarizes, and in this way spikes are produced. So in this way action potential is produced in the form of spikes, and that's why it is known as spike potential. So now we are very clear that slow waves play a very important role in the production of this spike potential. There are actually two types of contractions that occur in the gastrointestinal muscles, and these are tonic contractions and phasic contractions. Prolonged persistent contractions of smooth muscles are called tonic contractions, and the contraction which is followed by a relaxation, then contraction, then again relaxation, then contraction is known as phasic type of contraction, and spike potential determines this phasic contraction. In this way, resting membrane potential can be made more negative or it can be made more positive. So the factors which make resting membrane potential near 50, these factors will excite the smooth muscles because more of the slow waves will go above the threshold potential level. So muscle contraction will be increased, and the factors which cause the resting membrane potential near 70 will decrease the excitability of smooth muscles. And when excitability increases, then more of the slow waves are above the threshold potential and spike potential is produced more. And when resting potential becomes 70, then most of the slow waves cannot reach above the threshold potential and excitability is reduced. So we should know that what are the factors which increase the excitability of this membrane and make resting potential near to the threshold potential. One of these factors is stretch in the smooth muscle. As we know that membrane of smooth muscle is elastic. So when it becomes distended due to the presence of food in the GIT or any other factor, then calcium channels will become wider, and therefore more influx of cations will occur, and this will cause excitability, and this will cause increased excitability, and in this way action potential will be produced. Other factor which can increase the excitability or produce the action potential is acetylcholine, because it stimulates the receptors to load more cations in the muscle, and in this way it causes the excitability and increase in the action potential. The third factor is parasympathetic system and some hormones. As the production of action potential causes the contraction of smooth muscles, so this production of action potential is also under the control of parasympathetic nervous system and under the control of some hormones. There are also some factors that decrease the excitability and don't contribute in the production of action potential. These factors are the stimulation of smooth muscle cell by norepinephrine and sympathetic nervous system. So norepinephrine and sympathetic nervous system decrease the excitability and they don't contribute in the production of action potential.